Chapter 3 looks at the cell. We look at the cell, all of its parts and pieces. Again, this is something that you should have had prior to this class. So hopefully there is a lot of review here. There's mitosis. Obviously, these are going to be parts and pieces that we talk about in very general terms, and we'll slightly change their names a little bit depending upon the system. And, I'm hopefully on the side of you guys are not shocked by any of that. So today's lecture, hopefully we get through it all, we'll see. And this is hopefully the pretty much last huge day of review in terms of the content. At least I think so. Maybe a little bit with enzymes next week or next class. But hopefully this week will be the last of the kind of review content material. So without further ado, let's look at the cell, and we're going to start on the outside and work our way in. It is the plasma membrane is where we're going to start this journey. Why are cells so important? Because they are the basic building block of every system that we're going to be seeing. It is the shape of the cell, what the cell has inside of it, organelle-wise, that is going to help dictate a lot of the functionality that we're going to be talking about. When we look at a cell,
Dr. Thornley, I'm sorry.
Yes.
For some reason, I don't know if it's on my end, but I can't see your screen, and every couple minutes on Zoom, it kicks me out to join a meeting. I don't know if I'm the only one seeing that, but it kicks me out of the meeting. I don't know if it tells you if I'm leaving or not, and then it puts me back in, but I can't see anything.
Can anybody else? Does anybody else have that issue?
No.
No.
Okay. Log out and just restart whatever device you're using and see if that helps. And who knows?
Sorry, it logged me.
All right. I don't even know. Oh, yeah. 1, 2, 3 basic regions that we have with a cell. We have the boundary, plasma membrane, defines the shape of this sucker, separates the out from the in, separates the in from the out. It's going to help us to establish resting membrane potential for nerves, resting membrane potential for the muscles in terms of how they function. So they do a lot of fantastic things. The organelles and the cytoplasm that they live within, fluid and the little organ parts of a typical cell, and obviously Mr. Nucleus, which does all of the thinking for the cell, directs all of the activities. Yeah, okay, yeah, and there's a typical cell. A nice little summary. All the little fantastic organelles that we have. Be a fantastic slide to be looking at when you're reviewing for this material. All right, so let's look at the plasma membrane because that's what we want to start first, and we'll just work our way in from there. It is a phospholipid bilayer. We have a fabulous hydrophobic center here. Hydra means water, phobic means fear of, right? And we have the little phosphate heads that love to be in contact with the water, and they maintain this orientation all the time. It is when stuff fuses with the plasma membrane, we maintain this bilayer, this phospholipid bilayer. All of the organelles are phospholipid based as well around them. So when they fuse with this sucker here, I can maintain the integrity of that plasma membrane. If I lose integrity of the plasma membrane, obviously the cell will die. Okay, so I got to maintain the viability of this plasma membrane to maintain the viability of a typical cell. And you can see we have a lot of fantastic things in there. We have proteins. Sometimes we have proteins only on one side. Those would be referred to as peripheral. Sometimes they could be on the outside only, on the inside only. Doesn't matter. It's always peripheral. Sometimes we have them spanning the entire width of that plasma membrane. Like there's peripheral only on one side. Here we have this integral because it's integrated within the phospholipid bilayer. These things can move around. There's a lot of fluidity to this thing because of, right there, cholesterol. Cholesterol provides some fluidity to this plasma membrane. They are not stiff. They are not rigid cell membranes. And they shouldn't be. They should have some fluidity, some bendability, and cholesterol is what allows that to happen. So cholesterol is not a total evil thing. We need it. It is selectively permeable, so don't only let certain things through. Okay, stuff that's small, stuff that is not polarized, stuff that can dissolve in lipid membranes as well. Some of the functions, and we'll highlight, we'll talk about a little bit about structural support, but for the most part for this class, I'm more worried about transport as a function of plasma membranes. Excuse me. They're going to be a major thing in terms of bringing things across. Sodium ions, potassium ions, hydrogen ions, chloride ions. We're going to be looking at specific transport mechanisms, glucose molecules, all that fun stuff. They're going to be involved in helping to regulate a lot of processes through enzymatic control, bringing certain things across. Neurotransmitters. Our world is our oyster when it comes to what we can bring across with these proteins that are found within the plasma membrane. Receptors. We have a number of receptors. Some are specific for neurotransmitters that then open up a transport protein within the plasma membrane to bring certain things across. Self-markers. Well, one of the most important things when it comes to mounting an immune response. Right? We have markers on the surface that say, "Hi, I'm you, don't kill me." We have markers on the surface that is not us. And that allows us to know that it is a foreign invader and we can attack it. And we can get rid of it. All right. So a lot of functions here that we're going to be talking about at various different points throughout the semester here. Again, there's that cholesterol that I talked about there. Again, flexibility being its most major function here. All right. Most major function. But also we have carbohydrates attached to a lot of stuff. We have carbohydrates attached to proteins. We have carbohydrates attached to lipids, glycolipids, glycoproteins, and they're involved with recognition a lot of times. So you're going to see them in an immune function. So they'll serve as antigens, markers that allow you to figure out it's you and not a foreign invader. They're also going to be involved with some various different regulatory mechanisms in terms of getting white blood cells to stop and get inside. So mounting an immune response, any type of inflammatory situation, if there's damage, some glycolipids will show up or glycoproteins will show up on the surface and notify the body that, hey, there's a situation here and I need somebody to come here and fix it. So carbohydrates can attach to a lot of other biomolecules like lipids and proteins. And they could do a number of various different functions. And we'll hint at some of these regulatory molecules when we get into the nervous system in the next unit. Since a plasma membrane is a boundary, is a barrier, it's going to be having a major role in obviously what comes across. We have certain things that can come easily across. They can diffuse easily across. But we have to have a number of other mechanisms because the vast majority of substances aren't small, okay? Or they have some kind of, They're not lipid soluble, but we need these things to come across. That's where we have a number of various different mechanisms to bring substances in. One of those mechanisms, again, this is a type of endocytosis, bringing in, is phagocytosis. Phago is cell, or cyte is cell, and phago is eating. So with this type of mechanism here, we are bringing in very big substances. It can also be referred to as bulk transport. And when it comes to the name bulk transport, really the only place we truly talk about bulk transport is in terms of capillary exchange. Okay. So we'll be talking about bulk transport in terms of the cardiovascular unit. This is a major way to get a large amount of stuff, and by stuff I mean fluid, nutrients, waste products, a lot of stuff to move out of the capillary, the blood vessel, and into the tissue. Okay. To do that, to move a large amount of stuff, I can't just have it all go through the plasma membrane. There's a condition associated with the digestive tract if that happens, and that's leaky gut. So if you have that leaky gut syndrome, that is where we have a massive amount of substances, fluid, nutrients, the whole shooting match, moving through and between cells. That causes damage. It stretches, it breaks down that tight junction that can exist or that does exist between cells to make a nice tight seal to form tissues. And if you have way too much going across because of some kind of inflammatory condition here, you can have that leaky gut syndrome. So there is a rhyme and a reason to how much it does allow. Bulk transport is that way to potentially limit that amount of substance, fluid, nutrients, waste, all that whatnot, from moving from one side of the cell to the other side, from the apical to the basal membrane side. We have some other cells within the body, namely neutrophils and macrophages, that will send out tiny little feet-like extensions called pseudopods and engulf. That's this fantastic picture right here of this forming around this little bitty food vacuole here. Okay. Macrophages are cell eaters. They will go and break down stuff that's not doing what it's supposed to do. It will go and kill foreign invaders, very generally, not very specific. And they'll do this same thing where they extend these feet-like extensions. It's just extensions of the plasma membrane that just wrap around. Okay? So if this is a foreign type of cell here, okay, macrophage comes up to it, and what it does, it'll start to extend those pseudopods around it. Okay. And it continues until it completely has engulfed around it and brought it inside, mixes it with some digestive enzymes, and bada boom, bada bing, it's gone. So there are a number of ways that we can help mount an immune response very generally, and that's what macrophages and neutrophils really like to do. Body defense, inflammation, apoptosis, What's apoptosis? Anybody know?
Isn't it like the destruction of cells?
Programmed destruction of cells. Programmed cell death. Skin between your fingers in utero. Cells that are not functioning properly. Organelles that are not functioning properly. Apoptosis is that term that we use, okay, for programmed cell death. Some other methods of endocytosis that we have, pinocytosis is quote unquote cell drinking. It's really more than just bringing in fluids. There are some small things that can come across, ions and whatnot, when it comes to this. So it's not necessarily just cell drinking. Okay. There are some smaller bits and pieces that can come in and come across that way. It's generally not very specific. Is that to say phagocytosis is specific? No, not necessarily either. A foreign invader is a foreign invader, whether it's a bacteria or a virus. So that's not very specific if a macrophage can just engulf anything and everything. What is specific, and we'll talk a lot about various different receptor mechanisms of bringing in very specific things, looking in especially the nervous system, okay, is receptor-mediated endocytosis. With receptor-mediated endocytosis, and I don't think I have a specific nice little fancy picture, but we have receptors that will coat a specific region of the plasma membrane. And they're looking for a specific shaped thing. Neurotransmitter, hormone, cholesterol. Okay. Something I need to bring in. Specific shape, admit. Specific shape, specific binding to the receptor can bring in the specific substance. So with receptor-mediated endocytosis, they're generally looking for something very specific, all right? Cholesterol is an example. Glucose is an example. Specific shape, binds to something with a very specific shape, brings that specific thing across, okay? So if you're looking at a major difference between these two things, it's really about specificity. Okay? It's really about specificity. And these are just endocytosis type of things here. Okay, there's the vesicle fusing within the cell. There it is. Okay, here's where it starts out. And that plasma membrane, just as this thing invaginates in, it'll just do what it do right there. And it brings it within. And I've maintained the integrity of that plasma membrane. I've maintained the integrity of that plasma membrane, and the cell doesn't die. Okay, same thing when this thing comes up here, it fuses with it, and it opens up, and it dumps its contents out. Again, maintaining the integrity of the plasma membrane. For everything that goes out, there's stuff that comes in. There is usually a nice tit for tat type of thing. All right. This maintains the size of the cell as well, right? As if I'm bringing in things, okay, and that vesicle, as we bring it in there, what happens to the plasma membrane? It's going to get smaller. Well, same thing if a vesicle fuses with the plasma membrane to dump stuff out, it's going to get larger. So there's always even flow of information, of stuff coming in and out of this cell at any given time, and that helps to maintain the integrity and size of the cell. Exit is exocytosis, removal from the cell. How it does that, we'll look at that, I think in chapter 6. but we have some specific proteins that are involved with that. And we'll look at it very generally. Okay, we'll look at it very generally. But it's those fancy things that, and it's kind of cool because, you know, we can then target, you know, various different medications and whatnot to attack those proteins and stop the release of stuff from a cell. So kind of interesting there with how exocytosis is. But exocytosis is just removal of stuff, okay? Waste products, and when I say waste products, it's just stuff that the cell doesn't need to use, okay? It's not for being used by anything inside the cell, okay? And a lot of times proteins are made inside of the cell and they are removed. They are released from the cell because they're going to do something else within the system at another location. And it's all exocytosis. We have a few different modifications, if you will, of the plasma membrane or extensions off of the plasma membrane. They are cilia, they are flagellum, and they are microvilli. Sorry, keep hitting the wrong button on my mouse. Depending upon where the cell is located, depends on what type of cilia it technically has on its surface. We have some cells within the nervous system in the special sensory structures. That is, if we look at the cell, just draw a typical cell, you're going to have this real long one and some shorter cilia. Okay. But you'll have this, what's called a primary cilium. And it has a sensory function, guys. It's going to help us to determine, you know, sound, where's sound coming from? Am I moving left or right, forward or backwards? So stuff like that. So there's a lot of sensory ones. Taste. You have a very long primary cilium within the taste buds there. And it's going to be involved with obviously detecting various different tastes. We also have cells that all of the cilium are much the same size, all long, all the same size. And these generally beat in some kind of wave-like motion going back and forth. And these are there to move substances. When you think of cilia, this is generally what you think of, all right? This is there within the respiratory tract, within the trachea, all right? We have a number of cells beating in unison, going in one direction. That helps to get rid of stuff, all right? When you're a smoker, you kill those things off. So that's why a smoker needs to cough to get stuff up and out, all right? We have a number of goblet cells in that respiratory tract as well, in the trachea there, producing mucus to trap any foreign particles that you inhale when you breathe. And that cilia helps to beat them up towards your mouth. Again, smokers destroy those. The great thing about it though, I mean, as soon as you stop smoking, within about a few weeks, these little cilia can regenerate. So pretty resilient things. Looks like seaweed, okay? Looks like seaweed there. And that's what it looks like on a light microscope, unspectacular. Looks really spectacular here, scanning electron microscope. When you think of flagellum, you think of Mr. Sperm. This is involved with propulsion. Okay? That's really the only one in the human body, and that's really the only thing it does. Okay? It's single, generally not a huge number of them. One sperm, one flagellum, and that's going to be involved with propulsion through that mind trap that is the female reproductive system. Okay? The last one are microvilli, and by the name, you probably have figured it out. They're really small. Brush border is probably something that you've heard it described. If you remember, well, you never looked at microscope slides there in the lab, but if you did, you would have seen a number of them that were labeled as brush border. Brush border just means tiny little hairs, okay? And they are there for really one thing and one thing only, and that is to increase surface area. You find them a lot within the intestines because, especially the small intestine, because that is where the absorption of nutrients is primarily occurring. And you find them throughout the various different kidney tubule structures. Again, there's a ton of reabsorption that's occurring within the proximal convoluted tubule and some of the descending nephron loop. And the two major functions of those major areas is going to be reabsorption of nutrients. All right. So they're there to increase surface area. So when you see them there, they're there for that purpose only. Okay. They're not helping to really move a whole heck of a lot stuff across the surface. They're just there to increase surface area so I can bring more stuff into the cell.